There you go.
There, that's the starting gun. You can go now, Mark. We've got 5 attendees here so far.
Okay. All right, let's so start now. Okay. Hello, everybody, and thanks for attending Rare New England's Genetics Career Fair. Rare New England, or RNE, brings together New England patients, families, and providers touched by rare and complex disorders. It offers educational opportunities, creates awareness of available resources, and builds foundations of support to improve patient quality of life. RNE also supports the educational and professional needs of healthcare providers. Help the providers, and you help the patient community. Regarding tonight's program, I would like to thank the following sponsors for their generous support of RNE's career fairs: the New England Regional Genetics Network, Homology Medicines, Ionis Pharmaceuticals, Ultragenyx Pharmaceutical, and Vertex Pharmaceuticals. So my name is Mark Horson. My specialty is pediatrics with a focus on genetics as a subspecialty, specifically the area of biochemical or metabolic genetics. So if you think back on your biochemistry, I deal with patients and families who have defects in those biochemical pathways. Biochemistry can be abstract and rather dry, but it is that patient contact that really puts the human back in biochemistry and shows how relevant the urea cycle or the electron transport chain really is. Why are we doing this program tonight? Well, genetics impacts almost every area of medicine. So as you can imagine, there's a growing need for geneticists in this country and abroad. Patients and families want and need to hear information about the diseases they have, information to plan their futures, information as a couple with a family history looking to have children, and information about effective therapies now available for genetic disorders. And considering that there are over 7,000 rare diseases, not all genetic, and growing, that's a lot of information that needs to be shared. But the impact of genetics is not just in rare diseases. Genetics can impact how we manifest a lot of mainstream diseases, including COVID, how we respond to disease, how we utilize medications, what medications are available or effective or safe, and so on. This evening, you're going to hear from 3 physicians who specialize in the area of genetics: a clinical geneticist, a general clinical geneticist, a biochemical or metabolic geneticist, and a geneticist who works in research. You'll hear a bit about what they're doing, how their genetics expertise has helped to treat their patients and families, and how the experience has impacted their own lives. A Q&A session will follow the 3 presentations. Please submit your questions as we go in the Q&A area, as opposed to the chat area. After that, I will guide you through additional opportunities to learn more about genetics. But first, you might think that liking biochemistry makes me a nerd, and the fact that I bring biochemistry into the clinic makes me even more of a nerd. Well, fun fact, I am a nerd, but we genetics nerds can have fun. Let me show you how with this very interesting genetic pedigree. And can you see that? Nope, that's not what I want. What do you nope. Oops. I am a little lost in this.
We saw it for a second, Mark. So you had the right file up, but then you changed it.
Okay. The wrong one came up. Okay, there we go. And here we go. Many, many years ago, when I was 23, I was married to a widow who was pretty as can be. This widow had a grown-up daughter who had hair of red. My father fell in love with her, and soon they two were wed. This made my dad my son-in-law and really changed my life. Now my daughter was my mother because she was my father's wife. To complicate the matter, even though it brought me joy, I soon became the father of a bouncing baby boy. My little baby then became a brother-in-law to Dad, and so became my uncle, though it made me very sad. For if he was my uncle, then that also made him brother of the widow's grown-up daughter, who of course was my stepmother. Father's wife then had a son who kept him on the run, and he became my grandchild, for he was my daughter's son. My wife is now my mother's mother, and it makes me blue, because although she is my wife, she's my grandmother too. Now, if my wife is my grandmother, then I'm her grandchild. And every time I think of it, it nearly drives me wild, because now I have become the strangest case you ever saw. As husband of my grandmother, I am my own grandpa. Oh, I'm my own grandpa. I'm my own grandpa. It sounds funny, I know, but it really is so. I'm my own grandpa. Written by Mo Jaffe and Dwight Latham. If you want, go onto YouTube, you will find any number of videos with that song, but you will not see the pedigree there. That is a special treat for you tonight. So with that behind us, I would like to introduce our first speaker. And this is Dr. Jody Hoffman. Dr. Hoffman is Associate Professor of Pediatrics at Boston University School of Medicine, and she practices medical genetics at Boston Medical Center, where she is Chief of the Division of Genetics. She also serves as Medical Director of the Boston University Genetic Counseling Master's Program. She received her medical degree at Albert Einstein College of Medicine in the Bronx. She then completed a pediatric residency at Yale Children's Hospital in New Haven. Her medical genetics training was completed at Children's Hospital of Philadelphia. She practices as a general medical geneticist and provides counseling, diagnostic care, and management regarding prenatal care, neonates, children, and adults with genetic conditions. She plays a role in the optimization of lab utilization of genetic testing, serves as a member of the Scientific Advisory Board for the National Tay-Sachs and Allied Diseases Organization, and aids in the release of incidental genetic findings for the Framingham Heart Study. Jody, thank you so much for being here this evening.
Thank you so much, Mark. And the part of that that was not announced was that I actually can credit much of my entrance into the field of genetics from the time that I spent with Dr. Corson when I was a undergraduate student and attended a camp that he ran for young ladies with phenylketonuria in order to keep them on their diet lifelong so that their children would be healthy. So this is not the first time I have seen him nerd out and sing funny songs. So we do have some sense of humor with our nerdness. Anyway, so today I'm going to tell you a little bit about my practice as a general geneticist. And as Dr. Corson said, I'm now at Boston Medical Center, and I've been there for almost 7 years. And prior to that, I was at Tufts Medical Center with Dr. Corson for about 11 years. And so I'll tell you a little bit about what I enjoy about genetics. So first of all, I wanted to talk a little bit about what types of patients come to genetics. So what kind of gets me excited and interested. We have a lot of children come with what is called dysmorphic features, meaning features of the body that are formed differently than expected, like the outer part of this ear, visible differences in how the body is formed, whether they be the hands, the feet, or other parts of the body, internal anomalies, like how the heart is formed or how the inner part of the eye or the ear is formed, unusual growth parameters, meaning a child is very small or very large or has a very large head or a very small head, unusual skin findings, whether that means unusual patterns like you see here or certain rashes, intellectual differences, developmental differences, autism, unusual behaviors, such as food hoarding or compulsion to eat, people who have seizures along with other issues like intellectual differences or how their facial features are formed, as well as a lot of people who come with a family history of genetic conditions or cancer in the family, or things that they really want to understand as to whether they have a risk for having these conditions and what one can do to keep them as healthy as possible for as long as possible. So part of what I really enjoy about being a geneticist is that we kind of act like sleuths, that we're always trying to solve mysteries for families. And so one case that comes to mind that was very meaningful to me was an 8-year-old girl who had been seen by multiple geneticists prior to me coming to Boston Medical Center. And she was coming because she had seizures, she was unable to speak, and she was unable to walk. And she had had a lot of testing in the past, kind of gene by gene, as well as groups of genes at a time. And this was quite remarkable for being completely normal and unable to identify the cause of her issues. But by the time she came to me, genetic testing had really become much more readily accessible through large panels of genes, like 300 at a time instead of one by one. So we were able to send off a sample of blood to look at a group of genes that are all involved in the production of seizures, plus or minus other conditions. And so this child was actually found to have an abnormality in a gene called ZEB2, which is important for a condition that causes seizures named Mowat-Wilson syndrome. And I actually, on reflection, felt somewhat sad that I had not recognized immediately that she had Mowat-Wilson syndrome, because I did have another patient that had had this condition. But as we have all learned in genetics and many other parts of medicine, is that much of the medical literature out there is based on pictures of people that are of previous European descent, as opposed to the much more diverse community that we see in America and certainly at Boston Medical Center. So this young lady was of African-American descent, and her facial features didn't immediately pop out to me as similar to those of another patient that I had seen with Mowat-Wilson syndrome. And the mother actually said to me at the time that we got the results that she was so excited to finally know what her daughter had, despite feeling that this was somewhat bittersweet, because she now knew that it was a serious condition and that we did not expect her to really get to walk and learn to speak fully, but that she was asking me, why is it that it took so long to get to this diagnosis? And so I was able to explain to her a little bit about the development of technology, but I also had to be honest with her. And we went on the internet and I showed her pictures of what pops up if you look at Mowat-Wilson syndrome. And you will see picture after picture of children of Caucasian background or white background. And they just didn't have the same facial characteristics as the child that I saw. So this mother has agreed to share this picture of her lovely daughter. And when I looked very carefully, the earlobes were pressed up a little bit. And that is something that we tend to see in Mowat-Wilson syndrome. But she did not have kind of the down slant of the face or the same middle portion of the face in terms of the nasal structure and a prominent nasal bridge. She had more of a depressed bridge, and she had more of an upslant of the eyes. And so this pointed out to me that I need to really think a little bit harder about the diversity of facial features across people of different ancestral backgrounds, and how I think about genetics. So not only I think was this, really important for the mother to understand why it took us so long to get to this point, the technology, as well as some limitations in our medical literature. But she was grateful at that point to be connected to family support groups and be able to talk to other families and other providers about what to expect next. So that was very rewarding for me. I also have a patient that I'll never forget, who is a young lady who has a disorder called pycnodysostosис. And this is a recessive skeletal dysplasia, meaning that her bones are formed differently than expected. So these are not pictures of the young lady that I saw, but these are some other folks with the same condition. I had been following her long-term in Fall River at a satellite clinic that I attended. And she really impressed me, as did her family. So her parents had become pregnant at a very young age, around one was 15 and the other was 16. And they had this daughter with this difference in her bone formation, and they really stuck together and supported each other and helped her grow up into to be a very fine young lady. She has had a lot of setbacks in her life because her bones are much more brittle than other people. And one of her favorite things in the world to do is to dance. And so she insisted on being in a dance class every single year. And she almost never made it to the performance stage because she would always end up with a fracture in one of her bones. but she kept with it every year and enjoyed her dancing. She figured out how to get all the connectors that she needed in her car to lengthen the pedals on the car so that she could drive, and raise her seat so that she could see, and really become a very independent young lady. She went to a very well-known college, did really well there, and went on to public health in New York. And she's about to graduate from graduate school. And she was even willing to speak to my children as they were looking at colleges, which was really nice too. So I think we often get to develop relationships with our patients that we see on a yearly basis or more frequently, and can really be inspired by their fortitude, their families, and the ways that they get through difficult times. I also had another young man when I was at Tufts who presented with what we describe as coarse features, meaning that each of the facial features is a little bit more thickened or wider than we would expect. A gibbous deformity, which is the part of the spine at the lower part of the spine sticking out a little bit, kind of curving outward, which is quite unusual for a small child. A lot of mucus in the nose, as well as the need for removal of tonsils and adenoids at 9 months of age. And he was eventually found to have a condition called Hunter syndrome, which is a lysosomal storage disorder. And being that we identified that condition, we were able to transfer his care to Children's Hospital, where they do have a metabolic team, and we're able to get him a bone marrow transplant. And he's now doing fabulously and spoke at one of our pediatric conferences and wooed and wowed everybody in the audience. So lastly, a little bit about what I do when I'm not in the clinic. So I thoroughly enjoy all of the time with my patients, but I also enjoy participating in groups that look into why things happen to patients, what's causing them, and helping define new syndromes. So this is just a reminder of a really interesting case that I had where a child had the lack of a nose called arrhynia, and she was found to have a balanced translocation, meaning she had all of the genetic material she was supposed to have, but it broke and reattached. And this group in the Boston area through the Harvard system was able to identify that right at that breakage point was a gene that was really important for the development of the nose. And they were able to model it in a zebrafish and show that there was a difference in how the nasal structure formed if that gene was not working properly. So that was really exciting and a rewarding moment to be a part of that. I also spent a lot of time helping sort out what is the best sort of testing method for something such as Tay-Sachs, which is a storage disorder that can affect children at a young age, and how to screen people for their carrier status. And I continue to work on Tay-Sachs through the National Tay-Sachs and Allied Disease Group, helping support their staff with medical questions. And I enjoy working as part of the American College of Medical Genetics on different committees, previously an ethics committee and now a program committee. I get involved in cases that are often quite important to the patients and their families, such as children with genetic syndromes that might induce fractures or bruising, and whether those are actually due to genetic syndromes or possibly due to abuse. And I also, as Dr. Corson mentioned, enjoy being medical director of a genetic counseling master's program, which is a 2-year program to allow people to become genetic counselors. And so I get to do a lot of teaching and work on the curriculum with these folks. So overall, I found that genetics has been quite rewarding, and that there are lots of different areas of interest that I can pursue on a daily basis. So I will leave that on to the next person.
Thank you so much, Dr. Hoffman, for sharing that with us. All right, on to our speaker, Dr. Stephanie Sackarau is a medical biochemical geneticist at Boston Children's Hospital and is a member of the Harvard Medical School faculty. She graduated from Vanderbilt University and then from the University of Miami Miller School of Medicine. Board certified in pediatrics, medical genetics, and medical biochemical genetics, she has been practicing for over 15 years. She was recruited to Boston Children's Hospital in 2015. There, she assists in the diagnosis, management, and counseling of children and adults with a wide variety of inherited conditions. As an experienced dysmorphologist and medical biochemical geneticist, she sees patients with developmental disabilities, congenital anomalies, genetic syndromes, and metabolic diseases. She was involved with the implementation and execution of expanded newborn screening for metabolic disease in South Florida. At Boston Children's, she is director of the Dr. Harvey Levy Program for Phenylketonuria, or PKU, and related conditions, and heads up a very specialized clinic for PKU therapy. She's also a provider for the lysosomal storage disease program. She is associate program director for the Medical Biochemical Genetics Training Program and faculty advisor for the Genetic Student Interest Group at Harvard Medical School. She's currently involved in many research studies with a focus on PKU. Thank you for joining us this evening.
Great, thank you so much, Mark. So I'm really happy to be here with you today to share a little bit about my experiences. And what I'd like to talk about today is my work with phenylketonuria, or PKU for short. So this is a kind of silly story, but the first time that I remember hearing about PKU, I was in high school. I specifically remember learning about PKU because my friends and I, we called it the P disease. And then we made a joke if somebody had to use the bathroom too much, we said that they had the P disease. But of course, PKU has nothing to do with urinary frequency, but that is my earliest memory of PKU. So I thought that was kind of funny that I now work a lot with PKU. So I fell in love with the fields of medical genetics when I was in medical school. I previously didn't know that it existed as a discipline. And it seemed to be a great fit for my interests. And so, so as mentioned, I trained in, well, I don't think that was mentioned. I trained in pediatrics, then medical genetics, and was boarded in medical biochemical genetics. And so when I first became a faculty member, I really enjoyed seeing patients with PKU because in part, you get them as a baby. You see them when they're 4 or 5 days of life. From newborn screening, and then you get to watch them grow up, and you can literally continue to follow them for their whole lives. I have people with PKU that I follow from babies to their 70s. And so it's really nice having that type of continuity and that long-term relationship that you have. So a lot of patients that I see in my general genetics clinic, I'm helping with their diagnostic journey. And I may see them periodically over time, but not on as frequent and consistent a basis as those with metabolic disorders. So I'm going to share my screen briefly. Okay, just one moment. I apologize, I can't see. Here we go. Now I can see my buttons. Okay, so I was going to start by talking about one of my mentors. When I came to BCH, I was really lucky to be able to work with Dr. Harvey Levy. So I think you heard the name of the program that I'm now directing is the Dr. Harvey Levy Program for PKU and related conditions. And so I wanted to mention that one of the wonderful things about our field in medical genetics, it is a rather small and close-knit community, and there is a lot of emphasis on mentorship. So I've been really lucky and continue to be really lucky to continue to work with Dr. Levy. But he's been involved with PKU from the really early days. He helped to lead the newborn screening effort, and newborn screening started in Massachusetts in 1963. And he was part of that from the early days. And he has been treating some of the patients that I now see since over 50 years ago. And I started, when I came to Boston Children's, I started working with him on a clinical trial that was called the PEG-PAL trial. And so this was a trial for people with PKU, and it led to the development of a drug called pegvaliase. So fast forward 7 years later, and so I'm now directing the Harvey Levy Program for PKU and related disorders, and a clinic for people on enzyme therapy that came out of that trial, which is called, the enzyme therapy is called pegvaliase. And so back to the program, we had an educational symposium that we held just before the pandemic in 2019 to sort of kick off this new Harvey Levy program. And you can see him in the middle of the picture. And so that was really exciting to be able to bring people together from all over the country and even outside of the country who are working in the field. So personally, some of the work that I'm most proud of comes from working with adults, in particular with PKU. So I didn't really get into PKU yet, but it is a metabolic disease that causes accumulation of phenylalanine, which is toxic to the brain. And if somebody is untreated, it causes intellectual impairment. If people are treated early and consistently, they are not going to develop intellectual impairment, but they can have challenges as adults if they go off of the diet later. So adults with untreated PKU have trouble with a lot of aspects of their daily functioning. They can have mental health issues, but there is a reversible component, and people have a higher quality of life, it seems, when they're on treatment. So I do find a lot of satisfaction helping people with PKU to bring it under control. So if somebody is on a diet for PKU, if they have classical PKU, they're only able to take around 5 grams of intact dietary protein in every 24 hours, which is basically like a slice of bread. So they're basically on an ultra strict vegan diet, but not a vegan diet because they can't have any protein, and they have to take a formula to make up for the protein they're not getting from the food. And to my understanding, the formulas don't taste great, though this has improved a bit. So this enzyme therapy really helped to bring people back to treatment and bring their PKU under control. Because with taking a daily injection, people can ultimately go onto a regular diet, which for many of our patients, they've never been able to eat regular food for their whole lives. So I personally am a vegan by choice, but I sometimes think that I am the vegan doctor who's probably got the most people to eat meat. And to me, it's, it's not about what their decisions are. It's about them being able to make their own decisions about what they eat and to have a normal lifestyle, which is definitely facilitated by being able to eat, since eating is such a social activity. But a lot of people who are on the PKU diet not only miss out on those type of aspects, but a lot of times they're not getting complete nutrition. So I'd like to share just a little bit about a few of my patients. So there's a young man that I've been treating who was late diagnosed with PKU. So he does have borderline intellectual disability, and he never had a normal Phe level in his entire life until the last few months. And that is when the enzyme therapy kicked in. So his parents just could not get over the difference in him with his mood, his general capabilities. And everyone in his life noticed the difference, this amazingly positive difference, including his coworkers. And he just got his driver's license. So his life is really changing as a result of the treatment. I also wanted to share about a woman in her 50s with PKU. And so she had been on treatment, as many people her age, until they were around 6, and then they went off of treatment. And getting back on treatment as an adult is really, really hard in terms of the dietary treatment. So she was treated with the enzyme therapy, and she felt such a life-changing result from bringing her PKU under control. She said that at work, everybody's noticing her work performance is better. She has so much more energy. She's not on the couch like she used to be. She became involved in her children's lives in a whole different way from before. And she told me visit after visit, her greatest wish is to meet the person who invented this drug that made such a big difference in her life. So at this conference, that person was a speaker, and she and Dr. Sarkisian were able to meet. And my patient actually presented her with an award after she spoke. So that was a really special time. So this is another picture from the symposium where we all came together. And so getting back to patients, so for some of them, it's getting the Phe level under control. We have some people who are on a diet consistently since childhood into adulthood, but then they can get a new freedom of eating what they want to eat. So I had an adult that could only take 5 or 6 grams of intact dietary protein from food a day. But with the new treatment, she was able to eat a completely unrestricted diet right before her wedding. And she was marveling how at her honeymoon, she was at an all-you-can-eat resort. And that was a first for her. And it was really special. So the last patient I wanted to mention is a woman who is in her 50s. And she was actually born before newborn screening. And her mother noticed that she had developmental delay when she was a baby and would not relent until she got a diagnosis. And she actually managed to get a diagnosis by 6 months old, which is really amazing. I would like to give that mother an award if I could, because I was just so impressed with that. So she basically saved her daughter's life. She saved her daughter's brain. And she has some impairments from the first 6 months of not being on treatment, but she's basically an independently functioning adult. She's married, she works, and she has just come back to care with diet. So she's on diet therapy for the first time now since she was 6, and she notices really tangible differences. So for one of the examples is that she goes to church, she goes to Bible study classes, and she never really understood them until now. So it's been a really, really fascinating field to work in. And so the victories can be large for some people, small for other people, but they definitely help our patients to live better lives. So I'm wrapping up, but based on my experiences, I applied for a grant to help to bring people back to care that were lost to follow-up. So some people were discharged from our clinic when they were 6, may not know that things have changed, that there's new treatments, there's new recommendations. And so we're going to try to, at a minimum, educate them, catch them up so that they can make an informed decision about whether they would like to try returning to treatment. So in addition to that, I've initiated a few other studies with PKU. One of them has to do with neuroimaging and neuropsych testing. We have one survey we're doing having to do with COVID vaccines for people with PKU on pegvaliase. And I'm also involved in a new drug therapy trial and a gene therapy trial for PKU. So basically, it's a really exciting time in the world of genetics and metabolism. And I am genuinely looking forward to seeing what happens next. So thanks so much for letting me speak to you.
Thank you so much, Dr. Saccaro. All right. Remember, if you have any questions for the speakers, write them in the Q&A section, and I'll pose the questions at the end. Our third speaker is Dr. Matthew Warman, who is professor of genetics and orthopedic surgery at Harvard Medical School. He also serves as the director of the Orthopedic Research Laboratories at Boston Children's. Dr. Warman received his Bachelor of Science in Engineering from Brown University in Rhode Island and an MD from Cornell in New York. He was fortunate to have received excellent mentoring during his scientific training with doctors Adele Bosky and Bjorn Olsen. His research focuses on patients with genetic diseases that affect the skeleton and vascular systems. He and his members of his lab contributed to the identification of disease-causing genes for a number of Mendelian disorders, including metachondromatosis, achondrogenesis type 1A, camptodactyly arthropathy, Coxa-Vara pericarditis syndrome, and osteoporosis pseudoglioma syndrome, and non-hereditary disorders, including Clove syndrome, Klippel-Trenaunay syndrome, and arteriovenous malformation. Dr. Warman is a member of the Medical Advisory Board of the Osteogenesis Imperfecta Foundation and the Scientific Advisory Board of the Fibrous Dysplasia Foundation. Dr. Warman, thank you for joining us this evening.
Well, thank you, Mark. And thank you for those of you who are interested in genetics. As I was thinking about actually what Stephanie and Jody were saying, I think one of the most rewarding aspects of clinical genetics and genetics in general is the ability to make relationships with patients and their families that are not hour-long or week-long, as might happen if you were working in an emergency room or on an inpatient ward, but potentially lifelong therapeutic relationships. Mark, you may remember one of the families we cared for together with glutaric aciduria back in 1986. And those children, now adults actually, who live in Chile full time, have come up to visit me and Karen, and Harvey Levy too, who was our mentor at the time that we cared for them. So now, almost 36 years later, I still am able to interact with patients who I met decades ago. So for many years, I was actually a practicing clinician, but for about the last 15 years, I've turned my attention primarily to running a research lab, to teaching medical students, and to doing administrative work on behalf of patients and scientists that study or have disorders of the skeleton. And I may just go ahead and share my screen very briefly as well, to just give you a sense of some of the conditions that we've been thinking about. Are my slides showing? I assume they are. So these are photos of 3 individuals who don't have a hereditary condition. So this is not a phenotype or features that can be passed on from parent to child, but nevertheless, these conditions are due to genetic mutations. This individual here, this is Joseph Merrick, who went by the sobriquet, the Elephant Man of Victorian England. And he ends up having, in all likelihood, a condition called Proteus syndrome. The gene for this is now known. This young woman, a patient at Boston Children's Hospital, seen in the Vascular Anomaly Center, has a condition called CLOVES syndrome, which stands for congenital lipomatous overgrowth with vascular, epidermal, and skeletal findings as well. I think you can see some evidence of the skeletal findings here with the enlarged toes. There's actually an extra toe, which I'll show you in a different image. And this condition also in recent years has been discovered at the genetic level, and it's due to a mutation in the gene PI3 kinase. And this photograph, an X-ray of a child that has overgrowth of the fingers called macrodactyly. I think you can appreciate the thumb, the second and third finger are overgrown with even some fusion of the skin between the 2 fingers, is also due to a mutation that cannot be passed on from parent to child, but nevertheless can cause massive overgrowth. And these 4th and 5th fingers are not undergrown, these fingers are overgrown. And what's remarkable is that not only are the bones overgrown, as can be seen here on the X-ray, but the nails are overgrown as well. So a mutation that is not hereditary is capable of affecting the growth of multiple different tissue types in the body. Now, scientifically, what I find fascinating about genetics is that this concept of genes that can only arise in a developing fetus and give rise to birth defects, yet not be passed on, was first postulated by Mary Lyon, who also postulated the idea of exon activation in mammals. But she postulated the idea of mutations arising in a developing animal that can give rise to birth defects that cannot be passed on nearly 61 years ago. And her hypothesis was proven 43 years ago by Dorothea Bennett, who the same year that the first human test tube baby was created, made a test tube mouse that was a combination of an animal with a lethal mutation and a wild type animal. And that allowed the lethal mutation to survive and become manifest in the mouse. And why did this animal survive? Because the mutant cells were presumably being nourished by wild type cells. And then from a clinical genetics perspective, the dermatologist Rudolf Hapley thought, gee, there must be equivalence to this phenotype that occurs in animals and can be recreated in mice. And he postulated that some of these skin disorders, incontinentia pigmenti or congenital nevus syndrome, would be the human equivalent. So the upshot of all this is that an embryo might start off with all working cells. One cell becomes mutant, and wherever the progeny of that one mutant cell ends up in the body can give rise to the malformative disorders that affect the skeleton, vascular, and adipose system. And if that mutant cell were lucky enough to find it to the germ cells, either the testes of a man or the ovaries of a woman, and get passed on, that fetus in which every cell is mutant cannot survive. So these mutant cells would be lethal if they were in every portion of the body, yet they survive because they're surrounded by healthy cells. And this is not new to genetics and malformative disorders. This is paradigmatic of cancer, right? Cancer is not in every cell of our body. When cancer arises, it might arise in lungs, in liver, in skin. And the only reason that cancer cells are able to survive is because healthy cells in the brain, in the heart, in the intestinal system are working to provide those cancer cells with oxygen, with blood flow, with energy. So these somatic conditions always occur sporadically. They're never inherited. They affect body and areas randomly based on where the progeny of those mutants first cells end up. They can be from mild to severe. If the mutation arises very late in fetal life, a small area of the body might be affected. If it arises very early on in fetal life, it might affect a lot of areas of the body. They can't involve the whole organism or even a whole organ because that would cause lethality. And it shouldn't matter if you're a boy or a girl because these mutations occur equally. From a scientific perspective, the other thing that's very interesting about these is that these mutations can cause a patterning defect. Here you can count there are 6 toes instead of the usual 5. They can affect the allometric or the proportional growth of, say, the left leg compared to the right leg. Look at how large this foot is compared to the unaffected foot on the other side of the body. And they can cause a vascular condition as well. So for the last 10 years of my life, I'd say, I've put a lot of effort into trying to understand the causes of these conditions by studying affected tissue from individuals, sequencing it to find the responsible gene, understanding how that gene is involved in a pathway, and if we're lucky, finding a therapy that might be able to remedy the problem. And although my lab has been lucky to contribute to finding the gene and understanding the pathway, we've been beaten to finding therapies that can benefit patients with these diseases. But frankly, we don't mind because patients will not really care who discovers the best cause for their disease or the best treatment for their disease, as long as the treatment can be discovered. So I think I'm going to stop sharing my screen there. And I'm just going to tell you that I think the fun of being a research-based geneticist is that the questions we answer can begin with patients. We can use animal models, molecular biology, cell-based studies to identify genes, to model the disease in a mouse or zebrafish or even in a sheep and try drugs that might benefit those animal models of disease that can be transitioned into human patients. So I've been at this probably as long as Mark, longer than Jody and Stephanie. I've been at it for 35 years already, and I still love what I do. It's fun to come in to Boston Children's every day because there are patients that I've known for a long time. There's new things to be discovered every day, and there's patients who really need what Stephanie, Jody, and I do to both benefit them and their families. So I'll stop there. Thank you.
Thank you, Dr. Warman. I'll ask all the speakers to turn on their videos. Again, we've gotten some questions ahead of the program, so I will start with those. But again, anyone who attending who has any questions, please write them in the Q&A section. Dr. Sakharov, how are your patient encounters different from other clinicians?
That's a big question. I think Jody and I probably see a lot of the same types of patients, but the patients that have metabolic disease are really varied. Some of them come to us through newborn screening for conditions that are known to be treatable. There are a number of them, around 40 something, that are metabolic, that are screened for a newborn screening. So we may pick them up as babies and, like I was mentioning, follow them throughout their lives to manage their metabolic disease. So that's one of the things that makes it distinctive. Another thing is our on-call is very different because we have children that have acute decompensating disorders. So some of the patients that we see can become acutely hyperammonemic, like they can have a high ammonia level that causes swelling of the brain. They can have a high level of acid in their blood, other types of metabolic derangements, low blood sugar. So part of the management that I engage in is acute management of sick babies, in addition to the chronic care and diagnostic journeys of many of the other patients.
Thank you. Dr. Warman, are there any promising technologies in the works that you believe will assist geneticists in the future?
There's incredible technologies in the works that are assisting geneticists in the future. And I stopped seeing patients about 15 years ago. And in the old days of seeing patients, most of the time we could not make a diagnosis unless it was an obvious disease that was very well characterized. And we would spend hours looking through textbooks trying to see if there was a patient that resembled the patient that we saw in clinic. And I wonder if Jody and Stephanie would have a different experience now, and that if they don't know what a patient has within 5 or 10 minutes of going into the room, they may send an exome, they may send a whole genome, they may get a result and say, gee, if I had 2 or 3 hours to really think deeply about the patient, maybe I would have come up with that diagnosis. But thank goodness that there's next generation sequencing that's facilitating diagnoses. Then there's matchmaker exchange. If you have a patient and you send a sequence and you get a variant and you don't know what the meaning of that variant is, you can post the variant and the patient clinical features on an exchange where anyone else in the world who might have a similar patient or a similar gene mutation can make a match with you. Or a scientist who's working on that gene and might have an animal model that looks just like your patient or has features similar to your patient can make a connection with you. So that's there. There's techniques for making mice. I think the new and zebrafish, this new discovery of CRISPR-Cas9 gene editing by Jennifer Doudna and Emmanuelle Charpentier. Based on, as Jennifer Doudna says, she just wanted to understand why bacteria avoid getting the flu. And in doing so, she discovered this incredible gene editing platform that bacteria use to kill viruses and that we now use to genetically engineer mice. So we can recreate in a sheep, in a mouse, in a fish within weeks, gene mutations that used to take years to model. So I think the technology is incredible. And if you like doing science and being at the bench, even though it's filled with a ton of frustration because it's a lot of delayed gratification, the tools at our disposal nowadays are so much fun to play with, and we can learn so many things. And again, the fun of what we all do is I think we all learn something new every day. It's not like being a banker and pushing papers or being a lawyer and checking that things are all kosher. Every new day is a new learning experience and a new interaction with people.
Now, let's not disparage other career paths, but okay. But yes, let's be pro-genetic. Dr. Hoffman, you told us about some uncommon diagnoses. What are some common diagnoses when working in medical genetics?
So I would say we see a large number of children who have a clinical diagnosis of autism or developmental delay. And part of our job is to understand, is there an underlying genetic cause that we can give information to the family in terms of what to expect, how to care for the child, if there are other medical issues that are attached to that diagnosis that we haven't yet uncovered, and what the reproductive chances are for that same issue occurring again in that family member. So that's extremely common. I'd say we also see a lot of children for conditions such as neurofibromatosis type 1, where people have a lot of brown patches on the skin and some within the irises of the eye, and sometimes can develop tumors over time. Things like Marfan syndrome or Ehlers-Danlos syndrome, which are disorders of connective tissue that may affect bones or joints or growth or heart structure, and really are multi-system disorders. So there are quite a few that I think people have heard of before. And those are kind of what we feel most comfortable and used to, but I think we're also quite lucky to have the educational excitement of learning about syndromes that we have not yet encountered in the past.
Thank you. Well, let's start. I'm going to direct this next question to Dr. Sakharov, but I'm going to open it to Drs. Hoffman and Worman after. How did you decide which type of medical genetics to enter? Clinical, metabolic, laboratory genetics are so different. How did you decide, and when do you decide that?
So originally I was really interested in children with developmental disabilities and genetic syndromes. But once I was in my training, I was in South Florida and we didn't have expanded newborn screening when I started. And so I was there for the whole implementation of going from screening for 6 different conditions to screening for around 40 different conditions. And so that had a big impact. And I thought it was really special to be able to do that kind of work. So then I decided to specialize in medical biochemical genetics so that I could see an assortment of patients, including those with metabolic disease.
And any thoughts, Dr. Worman, Dr. Hoffman?
Well, so I went into genetics because I knew I wanted to spend time in the lab and run a laboratory. And I naively thought that geneticists were like the Maytag repairmen. That a lot of people probably listening don't know who the Maytag repairman is. But the joke was Maytag appliances were so reliable, the repairman just sat around all day doing nothing because he never had to fix anything. So I thought naively that, gee, geneticists, what are the emergencies in genetics? But of course, as you've heard, there are a ton of emergencies in genetics, whether it's been inborn errors of metabolism with life-threatening elevations of ammonia or lactic acid, the birth of any child with a birth defect or an unexpected condition is an emergency for that family. And I think the trait that a lot of geneticists have is the ability to visit a patient at a bedside during a really traumatic, catastrophic time for them and make it appear as though there's all the time in the world to help them through this very trying time, and then be committed to doing that for a lifetime of that patient. So I'm guessing that Jody and Stephanie share that trait. I know Mark's got it.
Mhm.
And then as I was trying to think about a subspecialty in genetics, I wanted to focus on the skeletal system, brittle bone diseases, dwarfing disorders, and more recently, the somatic vascular and overgrowth disorders, because I wanted to feel as though I was expert clinically in something, yet not in everything, so I could focus on the laboratory too. I'll turn it over to Jody.
So with all due respect to researchers, I must say that I spent my early part of my exploration in laboratories, and I kept telling myself I wanted to research medical genetics, but it turned out that no matter what lab I was in and how excellent my mentor was and how interesting the project was, I always wanted to be one step closer to the humans behind those disorders. And so, I absolutely respect and recognize those people who have the patience to sit at the lab for hours on end. But I needed a little bit more day-to-day interaction with the patients. And in general genetics, I found that when I first did my training, which was actually in pediatrics, and these days you can actually train in pediatrics or internal medicine or family medicine before going into genetics, I was very set on working with children, but it turned out that over the years, I ended up spending more and more time with adult patients. And now I kind of do 60% peds and 40% adults. And I find that the diversity of issues and approaches between taking care of a whole family who has a baby, or a family who's pregnant, or an older person who has a condition that they may have passed down, that whole family approach and the different ages involved is very intriguing and important to me. And I really like the family approach to medical genetics, and being a general geneticist allows me to do that in a very broad way. And I never feel bored, and I'm always learning more and working with people with different disorders of different ages. So I like the diversity.
All right, last question. Dr. Hoffman, I would ask you to check the chat because someone has a specific question for you.
Mhm.
Dr. Sakharov, do you believe that there are any difficulties in this field that medical students or pre-med students should be aware of, or that aren't fully explained before pursuing genetics? Or what do you wish you knew before you went into this field?
Well, I feel really good about my decision. I think it's a wonderful field to be in. Doing the call for metabolism, especially at a busy hospital like Boston Children's, is really stressful. And so I didn't really love acute care. I didn't love the ICUs and the ED rotation. I mean, I liked them fine, but that wasn't what I wanted to do. But that is a part of what I do. And so, the need is there, and there aren't that many people that are expert in these conditions. So I'm happy to do it overall, but that was something that I probably didn't anticipate to that extent when I started.
Okay, thank you. All right. What I'd like to do now is tell you folks some other opportunities if you have an interest in genetics, other ways of finding out more information. Following that, we will have an evaluation. I ask you to do just a quick evaluation of this program. So I'm going to share my screen. All right, so additional learning experiences. One-on-one conversations with a geneticist. So there are obviously different types of subspecialists, general or clinical, biochemical, research, and or industry. So this is an opportunity to speak with the geneticist about their careers and about your interests. It's a one-on-one connection in which the focus can be on your career path. If you're interested, then we can put you in touch with a geneticist. It could be near you, or because everything is done online now, it doesn't have to be, but please prepare a letter of intent, what your questions are, and send us a CV and we'll connect you. The American Society of Human Genetics has a great career information section. It's information about career options at all levels of the academic ladder, whether you're an undergrad, grad, medical student, or postdoctoral. And there's specific advice for about a career, how to do interviews, how to negotiate positions, and so on. The website is there. We'll be providing you all these opportunities, and we'll send these out to you, so you don't have to take notes. An on-site elective. This is an opportunity to shadow a geneticist who's seen patients in clinic. You hear about the patient and disease and the concerning issues of the day. But arranging electives requires planning, like hospital security clearance. Again, prepare a letter of intent and send us a resume, and we'll put you in touch with somebody. One can also do an online elective, and this is an online counterpart to the in-hospital experience, where we hear from 2 or 3 patients about living with their disease, and then we learn a little bit about their diseases. It goes on for about 60 or 75 minutes. The patients are either live patients or they're videotaped, and each elective has a particular theme, like muscle symptoms or brain dysfunction. Let us know if you're interested. RNE conducts a Rare Disease Day speaker series. So Rare Disease Day is the last day in February. And so what we do is we bring patients and caregivers to medical audiences around New England during the Rare Disease Day season, so anywhere between January and March. The doctor provides a 50-minute medical overview before the patient or caregiver speaks, so that the audience has an idea of what they're talking about. This is what we did at the beginning of this year throughout New England. So it's all the states were included, with a range of mostly metabolic disorders, because I run the program and metabolic disease is what I'm interested in. So, but very interesting. And the American College of Medical Genetics Foundation has a Summer Scholars Program. This is a funded 6-week elective experience for first-year medical students. So it's limited to first-year medical students in the summer following their first year. And applications are made available in December and for the summer, next summer. This is generally done through the hospital, but you can find out more information at that link. And then finally, the Global Genes Rare Compassion Project, where the applicants that are accepted, generally medical students, are matched with rare disease patients and families. Applications are accepted year-round. The matches are made quarterly, and the applicants are eligible to submit an essay about their experience to qualify for the scholarship, and more information at that link. So with that, I'm going to close this program. I want to thank very much our speakers for coming and sharing some of their passion. And you've done a great job. Thank you so much. Kevin, thank you for organizing this. Can you put on the evaluation for our participants?
Yeah, thank you, Mark. My name is Kevin Fried. I'm the Vice President of Rare New England, and I want to thank the speakers as well, and for the participants who joined us today as we try to fill up the pipeline and with more geneticists so that we can help more rare disease patients. You know, you guys are helping quite a bit on both sides of this discussion. So let me pull the poll up here. It's a very short poll. If you could just take a second or so to give us some answers, that would be great. Okay, it looks like that's all the answers we're going to get. Thank you very much. Have a wonderful evening, everyone, and thank you once again.
Thank you. Have a good evening.
Thanks for organizing. Thanks for having me.
Good night.
Thank you.